All right, we've got this figured out now. Yeah. Yeah, so I think the story starts in the past 10 to 15 years where we've really witnessed a revolution in our ability to sequence DNA, which is the basic blueprint of life. The cost of sequencing has dropped tremendously, even more so than what Moore's law would have predicted from the very beginning, and that's opened up an astounding new capability for us. We're to the point where sequencing entire genomes of organisms is close to being routine. And now there are hundreds of thousands of genomes that are available. Last I looked, the NCBI has like 200,000 bacterial genomes and many more from other species. And this technology boom has allowed us to investigate things in unprecedented ways, in ways that we couldn't do before. These include investigating the basis of human genetic disease, including some of the ones that are shown here. But at the same time, I think it's opened up new questions in that while we're very good at reading through sequencing, we're not as good about writing and manipulating. And to be able to make changes that the user can specify in order to interrogate what the function of genetic variation is really key. And so there's been a lot of technology development in the last couple of years to get towards that point. I don't think we're quite there yet, but I'd like to talk about some of the things that have been done to get there. So the story really starts in 1987 when a group of researchers sequenced a tiny snippet of the genome of the bacterium E. coli, studying a mechanism that was a gene that was totally unrelated to CRISPR. But purely by coincidence, at the edge of their contig, they noticed an unusual and enigmatic set of sequences that look like this. In particular, there were sets of 29 base pair repeats shown in yellow here. And in between those repeats were sequences that had never been seen before and didn't resemble anything that had ever been investigated. And this was a big mystery, why this was there and what it was doing. It almost looks like a sequencing error or a replication error. What became clear over the years was that these sorts of structures are very common in bacteria, and it's now known that approximately 40% of bacteria have these. And later on, they were called clustered, regularly interspaced, short palindromic repeats, or short for CRISPR. A major breakthrough came about 18 years later when researchers realized that some of these sequences in the middle are not random, but are actually derived from mobile elements, including viruses that try to attack the bacteria. So this is actually an exact sequence that was analyzed as part of the study. And they found that this particular spacer between the repeats matches to the genome of the bacteriophage P1. So this led to the immediate hypothesis that this CRISPR array might be some kind of genetic memory system or immunological recording that allows the bacteria to respond to viruses that come its way. And through the years, that hypothesis has been proven to be correct. And an amazing amount of details about an amazing mechanism have been elucidated. So this is roughly how it works. So we start off with a bacteria here that's being attacked by this phage. This is an actual micrograph of it. And you can see here the phage tries to inject its genome, which is shown in orange. But through the action of the CRISPR array, as well as CRISPR-associated genes, also known as Cas genes, the bacteria is able to capture a little snippet of this orange DNA and ligate it into its own genome in a permanent heritable change. So the genome gets just a tiny bit larger. But this dramatically changes the way that it can respond to future infections. So when the virus comes back again the second time, the bacteria already has a little memory of the first infection, and together with other Cas genes, it can respond to the infection by immediately recognizing any match to foreign DNA and cutting it right where it is to inactivate the virus. So beautiful mechanism here. At the heart of all of this, so there's a lot of diversity of CRISPR, but the most famous CRISPR protein that's been studied is an enzyme called Cas9 that actually does the immunity, the defense function that interfaces with the virus. So Cas9 basically is a protein RNA complex. So the protein here is shown in this big blob here. And it uses a piece of guide RNA that's actually transcribed from the CRISPR array. The guide RNA has two regions, the blue here, which is the variable portion of the spacer in between the direct repeats, and the red portion, which is the same for every target site. And this is an amazing machine that actually goes and looks at incoming DNA, in this case shown in black up here, and tries to find an exact match between the target and the guide RNA in blue. And if it finds an exact match of 20 nucleotides, it actually unwinds this target site and makes a cut right where the red arrows are. It can do this only if the target site is right before what's known as a protospacer adjacent motif, or a PAM. This is a 3-nucleotide sequence, NGG, that licenses Cas9 to be able to cleave things. So basically, this is a single platform. The graphics are not showing up perfectly here. Okay. This is a single enzyme in which a lot of this stuff is the same, the protein and the guide RNA in red. But just by changing this variable region in blue, boxed in pink here, we can specify any target site in any piece of genomic DNA. And then Cas9 will just use this information to go where it needs to go and make a cut. And this is really powerful as a tool, because it's very easy to change this segment to reprogram Cas9 to do what it needs to do, while keeping everything else the same. There's one interesting thing else to mention is that in bacteria, once you make a cut, the DNA doesn't really get repaired, so essentially you just deactivate the virus. When you try to do the same thing in mammalian cells by artificially delivering the Cas9 into a mammalian cell, and then the cut happens, something very different happens, because mammalian cells have very sophisticated double-stranded break repair pathways. And instead of just making a cut and having the cell die, which is what happens in bacteria, in a mammalian cell, once the cut happens, a lot of host enzymes will interact with the double-stranded break and make little mutations right near where the cut happened. And when that happens, it becomes a very powerful way to introduce local sequence changes right near the cut site, which again can be specified by the guide RNA. So this is the backbone of CRISPR genome editing and everything that's been built from it. So CRISPR is more than a nuclease. So Cas9 has 2 nuclease domains, one at the beginning and one in the middle of the protein called the RUV-C and the HNH domain. Each is responsible for cleaving a different strand of the double-stranded DNA. But it turns out we can just make 2 single amino acid substitutions at each of the catalytic sites of these nuclease domains to get rid of all the nuclease activity. And when that happens, it actually becomes a programmable DNA binding domain. So if we keep the nuclease activity, it'll make the cut and bind to the DNA where it needs to bind. If we get rid of the nuclease activity, it can just bind there and recruit other things to local sequence of DNA that can then be very useful to do other kinds of manipulations. So lots and lots of tools have been built based on this, based on gene repression, activation, epigenome editing, chromatin imaging, like base editing. Like the list goes on, and it's well beyond the scope of this talk to really even touch on all of these things. So for the remainder of the talk, I'm going to specifically talk about using CRISPR to do genome editing. That is, to make precise and defined sequence changes to a stretch of genomic DNA in a living cell. So this has enabled a lot of diverse applications over the years. Here are some examples. These researchers made these dogs a lot more muscular by editing some of the muscle genes. Here are genome-edited plants. We can use CRISPRs to spread genes in wild populations of organisms in a technological advance known as gene drives, as well as in human therapeutics, muscular dystrophy, and sickle cell anemia, as well as targeting T cells in cancer immunotherapy. And beyond all of this, I think Cas9 is just a powerful tool to be able to systematically interrogate the consequences of genetic variation on a research level. So for the remainder of the talk, I would like to outline some nuts and bolts of doing a CRISPR experiment for genome editing in the lab and what some of those things look like in terms of the major considerations. So spend most of the time on talking about the design, and then if I have time at the end, a little bit about expanding the CRISPR toolbox in terms of recent research frontier developments. So I think there are really 3 things that I want to highlight about doing a CRISPR experiment in the lab. The first is choosing an editing strategy, depending on the kind of sequence modifications that you want to get. For example, if you want to get a point mutation, the editing strategy will look very different from wanting to get a big deletion or an insertion in the genome. And there's lots of different ways to do this, which I'll get into. The second thing is delivery. How do we get the CRISPR protein and the RNA, and perhaps a donor DNA, if it's necessary, into the cell? into the specific cells that we needed to get to in a high enough concentration that it'll work, that minimizes toxicity and minimizes off-target effects. And then finally, how do we assess how well it worked? How do we look at specificity and on-target editing outcomes? So to the best of my knowledge, there are 5 main strategies for doing Cas9 genome editing, depending on the sequence outcomes that one is trying to obtain. So the first and most, probably the easiest to do is just to knock out a gene by getting a mixture of small insertion and deletion mutations near the Cas9 cut site by a pathway known as non-homologous end joining, or NHEJ. This is basically just a cell responding to a double-stranded break by going into panic mode, trying to stitch together the ends and making mistakes in the process. And when it does that, that's useful to us. The second modality is very different. It's a system called base editing that was pioneered here by David Liu's group that instead of using Cas9 nuclease activity, it actually uses Cas9 to recruit a cytidine or adenosine deaminase to actually pull off an amine on a C or an A to directly convert a C to a T or an A to a G. So this is great for making transition single point mutations. We can also use Cas9 with 2 guide RNAs to make 2 breaks to delete a segment of DNA. So if a genomic DNA is cut into 2 regions with pretty high frequency, the stuff in the middle will just disappear. And then finally, if we're trying to make precise insertions of different cargo genes or just inserting based on a well-defined end goal, then we can use homologous recombination based on donor DNA or NHEJ-based ligation in a method called HIDI, which I'll get into. So this is strictly DNA level sequence edit. So how well this works in terms of what you need to do to get this thing to work well in your cell or organism of interest, there's a lot of variation in that. In terms of designing guides, how well guides work, there's also a lot of variation in that, and it really depends on which particular strategy you're going after. So I'll try to touch on some of the main considerations for each one. I should say that I ordered these roughly in order of difficulty. So the ones at the top are easier and more efficient, roughly speaking, not absolutely, than the ones at the bottom. Okay, so what are some principles for designing guides for single gene knockouts by Cas9 through indels? I think there's 3 main things to optimize for. One is high on-target activity. You want the guide to just cut really well. The second is to minimize off-target activity at potentially mismatched sites, so sites that have maybe 1 or 2 or even 3 mismatches, to avoid cutting more than once. And the third is editing outcomes. So usually for indels, you want a lot of frame shift mutations to actually disrupt the gene. It turns out how much frame shift you get varies by guide, and there are ways to predict that. So through a lot of pioneering work, guide prediction in terms of on-target activity and specificity is really quite sophisticated now. Through work by John Dench's group here at the Broad and many other groups around the world, there are now machine learning algorithms that can predict for each particular guide sequence how good it's going to be in terms of efficiency and specificity. There are a lot of online tools that allow you to just plug in a genomic target of interest, and it'll give you a list of guides and rank them by desirability. Here are some of them. I'd like to highlight one in particular. This is a tool from Benchling that integrates the state-of-the-art on-target and off-target scoring into one very nicely laid out form where you can just kind of read off the guides as they're ranked by each metric and test the ones that you want to test. So the other thing I mentioned considering is like what kinds of indels are actually created by Cas9. So it's often said that like if you make a cut here, say, you get a mixture of indels that's quote unquote random. Like you just get a random mixture of insertions and deletions. The picture that's emerging in the last year or 2 is that it's not actually random, and that the types of insertions and deletions here, whether it's like 3-base deletion, 5-base deletion, or a 1 nucleotide insertion, is heavily dependent on the local sequence context surrounding the cut site. So there are basically 2 main factors that contribute to this, as well as other less quantifiable factors. So the first is microhomologies near the cut site. So for example, if the Cas9 cuts here and there are just 2 repeats of the exact same sequence, even as short as 2 or 3 bases, that can activate an alternative repair pathway called microhomology-mediated end joining, in which there's a strong tendency to just collapse these repeats down into a single one. If the collapse leads to a frame-shifting mutation, that can be used to your advantage. But on the other hand, if this collapse is in frame, that can be a disadvantage. So paying attention to the kinds of microhomologies that are next to the cut site can actually make a big difference in terms of how much frame-shift mutation you're getting. The other factor is, surprisingly, the identity of the base 4 bases upstream from the PAM. So this base right here. It turns out if you have an A or a T at that particular base, there's a strong tendency to just insert another A if it's already an A, or another T if it's already a T. So if this is an A right here and you make a cut, then there's a strong tendency to just get AA and T to TT. So that can also be used to your advantage by picking guides that have an A or an T here. You can get higher proportions of frame shift mutations. So all of these, as well as other less directly quantifiable considerations, have been programmed and quantified by machine learning algorithms, and you can actually predict with reasonable accuracy the indel spectra of different cut sites. So here are some online tools that allow you to do that. Forecast, for example, you can just input the genomic target site, and it'll tell you roughly, like, if you cut at this particular location, what percentage of the time are you going to get a 1-base insertion? What percentage of the time are you going to get, an n-base deletion, and so on. So this can be useful for optimizing guides. Okay. Ooh. Okay. Get a sneak preview of So the second strategy, which is useful for installing transition point mutations, is base editing. So if you want to make a transition point mutation and base editing is a viable strategy for you, this is much preferable to any of the other strategies here. So if you can use this, it's a good idea to try to use it. And the way this works is that for a base editor, a Cas9 has actually been made catalytically inactive, so it just binds to DNA. And then there's a covalent fusion of different protein domains that actually deaminate either C to T or A to G. In this particular case, I've shown a picture of the A to G editor. So you have this covalent fusion here to an adenosine deaminase. So Cas9 will unwind the target DNA, and once this adenosine, this A, gets exposed here, the deaminase actually catalyzes this chemical reaction by hydrolyzing off the amine and then changing an A to an I, which is read as G. So this is a way to get this A to an I, which is G, without actually cutting anywhere in the genome. And this can be really efficient and much better than using homology-based methods. There's some limitations to this. So here are some editors shown. This is the C to T editor and an A to G editor. But really, the limitation is that the edited base has to fall in what's called this activity window. So some distance upstream of the PAM, NGG, and it's only edited efficiently if it falls in this particular window. So really, you just need 2 things for base editing. You need something that's sufficiently upstream from a PAM that places it right in the middle of this window. You need the PAM there. And you also need that particular edited base to be unique within this window. So if you're trying to edit 1 A, but you have 3 A's in this window, then they might all get edited, or some proportion of them, and that might not be desirable. But if you can find a guide that gives you this particular configuration, then base editing is very efficient. And a number of these constructs are available on AdGene. I should point out that I should point out that this is an active area of research, and a lot of improvements to the system in terms of shrinking or expanding the editing window and improving the PAM targeting range are active areas of research, and you can read more about it in Dr. Liu's review article out late last year. Okay, so the third strategy is to use two guides to make a deletion in the genome. A lot of this is very similar to the first one based on NHEJ, except you just have to deliver two guides. So a lot of the guide selection strategies have the same types of considerations. So here, if we deliver two guides, then the stuff in the middle tends to just get lopped out, leaving you with just the two flanks ligated to each other. Usually the junction is not totally precise. You can get little indels at the junctions. A good ballpark for how efficient this is if you're deleting a couple kb, then people have reported numbers of around 10% efficiency for getting biallelic deletions. So now I'd like to talk about the last two, which is actually introducing new stretches of DNA into the genome via insertions. So the first and probably the more popular one is homologous recombination. So this is similar to NHEJ, except instead of Cas9 creating a break, you also deliver a piece of donor DNA that matches the two sides of the break, but the stuff in the middle is different. And with some efficiency, usually lower than NHEJ, the cell can repair this break based on the instructions that you provide it in the donor template to precisely insert the stuff in the middle of the break that needs to get there. So for this, the guide design can be a little bit different because depending on whether or not you want the edits to be monoallelic or biallelic, the choice in terms of where you want to make the break relative to the insertion is very important. So here, if you want to get a biallelic edit, then it's important to make the break as close as possible to the intended insertion. Whereas if you just want to get one of the two alleles edited, then there's some optimal distance that you can pick, in this particular case, 10 base pairs to make the editing most efficient. Okay, and finally, the last strategy is NHEJ-based insertion. So HR-based insertion really only works in dividing cells in the S and G2 phases of the cell cycle. So if your cells don't go through those parts of the cell cycle, then it's hard to get this to work. But there is a very nice alternative, which actually just uses NHEJ to ligate in a big cassette. So this is roughly how it works. And for HIDI, you have a target site here in the gene of interest. And in addition to Cas9 making a break here, you also co-deliver a piece of donor DNA that has the same target site in the donor DNA. And when Cas9 gets in, it'll actually make a cut in the genomic DNA as well as in the donor. And with some efficiency, this will actually just get ligated in by the cell to create a final product that looks like this. So the end result is a little bit like HR, except it doesn't use any of the HR pathways, and so this can be much more efficient in non-dividing cells. Okay, so the next topic I'd like to talk about is delivery. So once we've chosen the CRISPR components and the RNA and the guides to put in, what's the most efficient way to get these in? So there are four main ways to deliver the CRISPR proteins, either via plasmid or a viral vector that's constructed from a plasmid. More rarely, directly injecting the mRNA. Usually this is only done for embryos, as well as direct delivery of purified protein RNA complex that can be just transfected into the cells. Usually, I think RNP is used most often for just creating knockout mutations in the cell. So in this case, this is much more efficacious than using a plasmid because you're not delivering exogenous DNA that could be, for example, toxic to the cell. A lot of these, for the first two options, plasmid and viral vectors, there's a lot of different great options on AdGene, and you can find lots of different configurations of vectors with all sorts of tags and things on them. Here are a few of our favorite general purpose Cas9. So PX330 expresses Cas9 in a guide RNA in a single backbone. PX458 does the same thing except with a GFP tag on the Cas9. 459 is similar except with a pyrimycin resistance cassette instead of the GFP. There's a lentiviral backbone that has both Cas9 and guide RNA, as well as another plasmid that has a smaller ortholog of Cas9 that makes it easier to package into AAV. And this is just a small snapshot of the many things that are available on AdGene. Okay, so finally, I'd like to talk about how to assess editing outcomes and making sure that the editing happened in the right way and that it was efficient. There are 4 primary ways I'd like to talk about doing this. The first one is if you're using HR or HIDI, for example, and you have a large-scale change to the genome, PCR genotyping is often just very effective in looking for big changes in band sizes, either in a mixed population of edited cells or if you grow out individual clones, you can screen cells that way. For indels and base editing, it's much harder to do it this way because the length of the sequences are not changing much, if at all. In that case, there are 3 main ways to assess editing efficiency. So the first is based on gel electrophoresis, in which you're actually just using a nuclease to cut heterogeneous populations of indels where they are. The second is based on Sanger sequencing, and the third, which is the most sensitive one, based on targeted deep sequencing. I'll just quickly go through each of the final 3. So in the T7E1 or surveyor assay, it is a way to assess editing efficiency for indels on a bulk population of edited cells in which there's a mixture of different types of indels. So we can start out with genomic DNA that has Cas9-derived edits. We just take the whole batch of cells mixed together and just PCR amplify the particular locus that's being edited. In this case, we get both amplicons in which there's no edit here, or there's some kind of indel mutation that's been acquired. We can take the PCR amplified products, denature them, and reanneal them, and they'll basically randomly reanneal so that you'll get some matched ones in which both strands have no edit, or sometimes both strands have an edit but the same edit. And sometimes it's mismatched where one strand has an edit and the other strand does not have one. If we incubate this product mixture with the T7 endonuclease, it actually specifically cleaves heteroduplexes like this right where stuff is bulged or mismatched. And if we take this and run this entire mixture out on a gel, we see something like this, where if you don't add the nuclease, you just get one clean band corresponding to all of these products. If you do add the nuclease, then you get one band corresponding to these products, and then some cut bands corresponding to this product that's been processed with the endonuclease. So just by comparing the intensities of these 2 lower bands with the top one, we can assess how efficient the editing was. This has a tendency to slightly underestimate editing efficiency because of this possibility. Like you might have 2 edited products that are the same that reanneal together, in which case you actually don't resolve them at all. Or they're similar enough, even though they're not the same, that you also don't resolve them. So the second way to do this is by Sanger sequencing. So this is very similar to the T7E1 assay in that we start out with a mixture of different kinds of edited cells. And then we PCR directly from that mixture to get a heterogeneous population of amplicons. But instead of treating it with a nuclease, we just take that entire mixture of amplicons and subject it to Sanger sequencing. So this looks something like this. So if there's no edit, then the trace looks all the way clean all the way through. But if there is an edit, then initially it looks very clean. But as soon as we hit the Cas9 break site, there's lots of mutations. And we see this jumbled mess of stuff that happens after that. It turns out that sophisticated deconvolution algorithms have been developed that you can just feed in this trace here, and it will tell you approximately what kinds of mutations and in what proportions are responsible for creating a mixed trace like this. So this is also a way to estimate editing efficiency. And then the third strategy is based on deep sequencing. So we can take the same pool of amplicons and attach Illumina adapters on each end, and then do single molecule sequencing on each one to say, does this have an edit? What kind of edit? And so on. So the pros and cons of these methods are as follows. So if you only have like a couple of samples, deep sequencing might not be the most feasible way to do it. We can typically detect maybe 1% to 2% indel with T7E1 or tide-based methods. The cost is roughly the same, but NGS is a lot more sensitive and accurate, and it can detect down to 0.1% indel or lower. But the cost of sort of having enough samples to merit this is a bit higher. I should point out that these two methods can also be used to assess the efficacy of base editing. So just like there's a mixed trace with Cas9 created derived indels, you also have a mixed peak at one position if there's a base edit that results in a mixture of AG or CT. And the similar thing is true with deep sequencing. Okay, so I'll take maybe 1 minute to just highlight some of the expansions to this toolbox that have been developed. So there are smaller orthologs of Cas9 that have been developed that are useful for packaging into viral vectors. There are variants with higher specificity that you can use to minimize off-target effects. There are variants that allow you to target different PAMs to expand the PAM targeting space in order to hit more sequences in the genome closer to the desired editing site. And there are other non-Cas9-based nucleases that have been harnessed from the natural world, including Cas12a and Cas12b. And you'll hear more about Cas12b tomorrow. Yeah, so finally, I'd just like to highlight that with our expanded knowledge of all the microbial diversity that's out there, this is really opening up a new frontier in terms of technology development, and there might be lots of other things, either in the CRISPR systems or non-CRISPR systems, that can be powerful to be harnessed for biotechnology, and that's something that we're interested in exploring further. So with that, I'd like to thank you for your attention, and I think we might save the questions for the breakout sessions. Is that right? Or? Oh, we'll take a couple. Okay. Yeah, so thank you very much for listening.
Thank you, Linyi, for that introduction. We can go ahead and take a few questions now, but you will have time to get more questions answered later. So just any quick questions for Linyi while he's still here? Yeah. Okay. Well, thank you, Linyi. We'll see. Just a second. Julia just ran off. So.
Can I ask a quick question?
Yeah.
So how often you see a large deletion for a single Cas9 generator breaks?
A large deletion for a single break.
How often you see, like, what's the frequency? You get small indels, right? But how often you see a large deletion?
With a single break?
Yeah.
We have not assessed that ourselves very quantitatively. I would say that overall it's not often. The indel spectrum for insertions, usually it's either a single base insertion or a deletion ranging from 1 to 20 nucleotides. We see a few that are larger than that. There's a paper out where they demonstrate that there can be the possibility of larger deletions and rearrangements that are not detected by PCR. So if the deletion site actually deletes one of the primer binding sites, which we put maybe 200 base pairs away, then we won't detect that. So I think probably the paper, I forget exactly which one it is, would be a good reference for that. I don't have a good number because we haven't explicitly profiled that ourselves.